Just don't accept it. It's just the best mechanism of running a business organization. The closer you are to that, the better institution you have.
Okay, good morning to everybody. Good morning and welcome back. Our first speaker today is Geoff McLachlan, and he will talk about clustering of time course chain expression data via mixture regression models.
Good. Thank you, Sylvia. First of all, I'd like to thank the organisers for the opportunity to participate in this workshop, and to thank you all for getting here at a comparatively early time in the morning for the Institute. I've got a joint appointment between mathematics and the Institute for Molecular Bioscience, and we have a relatively new building, so you should come to Australia and come visit Brisbane, and the bioinformatics section's on the 6th floor there, so there's plenty of room if you want to spend some time in Brisbane, in a relatively new building. Oh, I should say this is joint work with a former colleague at UQ Angazing and a present postdoc, Sam Wang. So I'm going to be looking at time course data, and with microarray technology now we have the ability to measure the expression levels of thousands of genes simultaneously, and of course a single microarray only gives you a snapshot at a given point in time, and people are interested, of course, in looking at these problems dynamically, and so there's lots of work being done recently now, or in recent years, on time course data. There's lots of papers being written, and in today's talk I just really want to focus on the analysis of gene expression profiles consisting of short time series of log expression ratios. So I've got a very modest goal. I just want to cluster these short time series. So I'm not trying to find a needle in a haystack or doing these tracking problems, some of the problems that people have talked on in earlier talks. So I'm going to be really concerned with clustering the gene profiles, and the biological rationale behind this is that if genes are co-expressed, then there's a chance they'll be co-regulated. And I suppose if you get a small cluster of genes and you know the regulatory mechanism for some of those genes, one of these first hypotheses that you might put up would be that all the genes in that cluster have the same regulatory mechanism. So, but we can also use the clustering to assist in functional annotation of the genes, and also there's always a lot of missing data, and so we can use the models to impute the missing data. So the outline of my talk, I'm just going to say something about mixture models in a general context, and then move on to the modifications where we've got, in particular, structured data here, because we've got data where there's replications on a single entity, and also we could have different classes. We could have sort of treatments, controls, etc. And then I'm going to actually talk about mixtures of linear mixed models, and then the clustering of gene profiles. So here's just an example here where we have some profiles, and if we think of this as being time along the x-axis here, then we just want to cluster these profiles into a small number of into a number of clusters. And of course you might say, well, why don't we just use Euclidean distance if we want to get profiles that are close together, and use k-means or something, but k-means will not take into account the order in which these measurements are made. It doesn't take into account the different sources of variation, and so we think we can do a better job in allowing for that variation, for the various sources of the noise. So, well, finite mixture models, that they've been extensively used. I think there's no need really to say anything more about their usefulness, and I think everyone is familiar with the definition, and having postulated a finite mixture model with G components, we generally take these components to belong to some parametric family, and then we fit the model by maximum likelihood using the EM algorithm. We have to do this iteratively, so this superscript k here denotes that we're on the kth iteration, and we form this posterior probability that the jth observation, or the jth member of the sample belongs to the ith group, given that you've got this observation on it. And then as k gets large and we get convergence, we can make a final, either we can do a soft clustering or an outright clustering. And this Zij or Zij, one can think of as a cluster label, and it's 1 if the jth observations is put in the ith cluster. And so we can estimate that by working in terms of these posterior probabilities. So this would be familiar, I think, of everyone on mixture models. Before I go on, I'd just like to go back in time a little bit. People do say that it's a sign of old age that one wants to dwell on the past, but I think it's this paper here by Nick Day. There's a nice Australian Cambridge touch here, because Nick Day wrote this seminal paper in Australia at the Australian National University. Unfortunately for Australia, he didn't remain there. He went back to Europe, and after some initial time in France, I think at the International Agency for Cancer, for Research on Cancer, he ended up in Cambridge here at the MRC Biostatistics Unit, where he spent the rest of his working life until he retired about 5 years ago. And John Wolfe at Caltech at the same time, they basically wrote down the equations, and they were astute enough to manipulate them into the form that you get it as if you just applied the EM algorithm. But of course, it was really the EM algorithm that gave thrust, brought on the use of mixture models, and I know I wrote my paper very shortly after that in '78, and then the books started to appear. David Hand and Brian Everett had a very small book out, and then Mike Titrington, of course, had a very comprehensive account with Adrian Smith and Yuri Markov of mixture models, and since then there's been quite a few books. I think at least 6. I probably might have missed out 1 or 2, but the other reason why I've mentioned those references, because when I read papers on mixture models by statisticians, and not necessarily people in machine learning, I'm not too sure what they reference, but the statisticians tend to basically only go back to the 90s. So if you didn't know anything about the literature and you're reading it, you'd get the impression that the initial work was done in the 90s, whereas it was really done by Nick Day back in, and John Wolfe, 40 years ago now. So I'm going to be talking about normal mixtures, and of course the attractive feature of that is that they're affine invariant, so it doesn't matter about changes in location or scale or rotation, but I think it's important, to have invariance under location and scale. And so here we are at the particular data we're talking about today. We've got these series of microarray experiments, and given that it's time course data, this microarray will be formed at a certain time, and then we've got at time 2 we've got this microarray, etc. And so these are these profiles that I wish to cluster. And so we could have a very large number of genes, but usually when we do the time course, we've done some filtering and selected genes that we know have got a certain period, well, they are periodic. And but in general, n is very large relative to capital M, the number of tissue samples. So if I could just put the problem of clustering the gene profiles in context, there are 2 clustering problems here. This one where we want to cluster the tissues, this is certainly a non-standard problem, because n, the number of observations, is relatively small, because there's not that many arrays in most studies, whereas little p, the number of variables, that's the number of genes, is very large. And so this is the big P, small m problem that has been the focus of this program at the Institute for the last 6 months. This problem here, well, it looks as if at first sight that it could be a standard problem, but not all the genes are independently distributed, and there's structure on these tissue samples. That there could be different classes, that there's replicated data, and there's correlations between the replications, and maybe among the genes. But if we just focus on clustering the tissues for the moment, if we assume a normal mixture model, we've got this covariance matrix which is P by P, it's highly parameterized, and we can perhaps do various things to try and reduce the number of parameters and to avoid having singular estimates. And Jeff Banfield and Adrian Raftery have looked at a spectral decomposition of these covariance matrices, but basically if P is going to be large relative to N, then basically you can't use that approach, because you just can't get reliable estimates of the full covariance matrices. So if you're going to just fit a normal mixture model where you just allow the covariance matrices to have their arbitrary form, a full form, then you're in some form of trouble, and you need to use some form of dimension reduction and or regularization. And our standard program for fitting mixture models is called mmix, for EM-based mixture analysis. It's very hard trying to find a name of software, because originally there was a program that Peter McDonald wrote with Bartlett called Mix, and then you've got Normix that John Wolfe had, Multimix that Murray Jorgensen had, M-Plus, Adrian Raftery and Chris Fraley, M-Plus, Nmix, Snob, AutoClass. So it's very hard. So we came up with Emma. I suppose people, perhaps my age, think of Emma Peel and the Avengers, sort of seem to have some a good name. But a lot of people call their software Emma, and I think MATLAB's got a well-known program called Emma. So anyway, we sort of started with this mmix. Now that's for when you've got conventional data. So when you're trying to cluster the tissue samples and P is very large, then we call this program mmixgene because the genes were the variables. And in this particular case, we've sort of got a three-stage approach where we look at the variables, the genes, individually, and throw away genes that don't seem to have much potential on their own, at least, for clustering. And then if we've got too many genes left, we cluster those genes into groups, and then work with some representative members of those groups, like the first principal component or the means, and then we cluster those representatives of the groups, so using mixtures of factor analyzers. So anyway, that's the problem that I'm not talking about today. We've got this other problem where we want to cluster the rows of this matrix, that is the gene profiles. Now, normally when we go to do our clustering using a standard statistical method, we've got really no replications that we know of, and we're assuming that all the observations are independently distributed. Now, in image analysis, where there's a spatial continuity, spatial correlations, we generally take it's common to take the observations to be independently distributed given the class labels, and we introduce the dependence between the observations by using a Markov random field on the class labels, like Julian Bezag did in his algorithm. So, so if we do have some structure, for example, if we had two classes and we want to cluster the gene profiles, then we could introduce the fact that we've got two classes here by using a regression model, and we could take our covariance matrix and then put this, take it to have this block form here, where we're allowing for correlations within the same class. But when you do that and you try to run your M-step, the solution won't exist in closed form, and it gets a little bit, I think, tedious. So I think the way to go is to specify what's going on through mixtures of linear mixed models. So, well, having called the program mmixgene, we'd really like to call this one MX gene, but we couldn't because we're clustering the genes. So we've called this one MX wire, where the wire stands for with random effects. So, and this one, came out in a paper in Bioinformatics in 2006. So here's our mixed model for the ith component. We're saying that an observation vector in the ith component has this, these are fixed effects. X is a design matrix, so is U and so is V. This is a random effects term to allow for correlations between observations made on the same entity. So if you're following a gene over time, this random effects term will induce correlations between those observations. Now this term here allows for genes that are put in the same cluster to be correlated. So this is where our model differs from most of the other models around, is that the presence of this random effects term here means that genes that get put in the same cluster will be correlated if the variances of this term is sufficiently large in the ith cluster. And then we've got this random effects term. Now, the presence of this term means that we can't write down the likelihood in closed form, but with the EM algorithm, if we take the random effects here to be missing data, along with the cluster labels, we can actually write down the complete data log likelihood, and we can actually do the E and M steps in closed form. So we don't have to resort to a Monte Carlo E step or anything. We can do everything in closed form. And after we've fitted our model, when it comes to trying to assign these observations, we condition on these cluster effects here, because once we do that, we can actually write down an expression for this density, which is simply a normal. It's a multivariate normal with this mean and this covariance matrix. And we do our final assignment of the gene profiles on the basis of those posterior probabilities. Now, there's lots of papers. I can't keep track of the papers coming out now on time course data, but Gilles Soler had a paper out, and our model would reduce to this one here if we ignored the random effects for the clusters. And more recently, Jim Booth and colleagues has a paper where he does a similar thing to us in introducing a random effects term for the clusters. So to demonstrate what we've been doing, we're looking at some yeast cell cycle data from this paper here of Cho. We've got 237 genes, 17 time points, and they would categorise these genes into 4 functional groups according to this classification here. And so, okay, we're going to have a go at trying to cluster these 237 gene profiles. Although they actually use oligos here to get to measure the expression levels, and the actual expression level is actually the log of the ratio of the intensity for a gene over the average value for that gene. So here's our linear mixed model. We're going to use this simple periodic function here, consisting of just cosine and sine terms, to model the periodicity in this profile. And capital T here is the period. And we've got these 2 terms here which can determine the time at which these genes take their highest and lowest expression levels. Now, for a given gene J in the ith class, all the measurements over time get this random effect term here. So that allows for correlation in measurements being made on the same gene. And at a given time in a given class, all the genes in the ith cluster get this term here at time 1, and they get this term here at the last time point. So that induces a correlation between genes that are put in the same cluster. And we can fit this model by maximum likelihood. So this is a model we're assuming in the ith cluster. We've got this fixed effects terms here. We've got this random effects here to allow for correlations over time, between observations over time. This is for correlation, to induce correlations between genes put in the same cluster, and then we've got that error term. Now, before we actually fit that model, we've followed the procedure, what's generally done in the literature, and that is we estimate the period first using least squares. So this is 0 here because the initial phase is 0. So we use least squares to get an estimate of the period here, which came out to be about 73 minutes. So we actually give an initial value to t, capital T. We then apply least squares. We then work out the mean square errors of the estimates of these betas. Then we increase capital T, and then we look at the mean square errors for different levels of capital T. Now, we actually can't write down the likelihood for this particular problem. So if we ignore the correlations between the genes and work on that basis and use the Bayesian information criterion, then as we increase G, we see that the BIC falls off at G equals 4 and then starts to increase again. So this would suggest just, of course, regularity conditions don't hold here for BIC to be valid, and we're also ignoring the correlations between the genes to get a likelihood, but it does suggest that we've got 4 components. And remember that we've got these genes were categorized into 4 functional groups. The other thing is, well, I should say that in fitting this mixtures of linear mixed models to lots of data sets, we generally get these variance terms here for the cluster random effect to be very small, and essentially we don't need it. But in this particular data set, in the first cluster, the variance comes out to be 0.23. So this is proportional to the correlation between genes in the first cluster, and this is proportional to the correlations between genes in the second cluster. So at least in these 2 clusters, it does seem that we need this particular term here. Now, so what we did is we compared the clustering we got with the grouping of the genes using the functional annotation according to the MIPS database. And so we used the RAND index and the adjusted RAND index to compare them. So this is our solution here, model 1. The RAND index is 0.7808, and if we permute the class labels to minimize the error rate when we compare our clustering to the functional grouping, we get an error rate of about 30%. Now, if we ignore the random effects of the clusters, that is, we take the genes to be independently distributed, we do actually get a smaller RAND index and a higher error rate. And in this particular model, we don't use the sine and the cos, we just fit a constant term at each time point. And that gives us a smaller RAND index and a higher error rate. And there's this recent paper by Wong, and she compared her method. She had a 2-stage method of clustering where she decomposes the distances between these profiles, a bit like you would in an analysis of variance decompose the sum of squares. And she compared her method to a lot of other clustering methods and found it to be superior. Well, she's getting her RAND index is lower than these. So anyway, for this data set, we were happy to see that the largest value for the RAND index came up for the full model. So as I was saying, the use of these random effects terms for the clusters seems to be giving closer result with the functional groups. So here are the 4 clusters we get using our full model, and if we forget about the random effects term for the clusters, that's what we get. And so, these are the 4 clusters when we use our full model. These are the 4 clusters when we don't use the random effects for the clusters. And it's a bit hard really to compare each one, but and this is the profiles grouped according to their functional annotation in the MIPS database. So here we have the 4 clusters using the full model, and here we have them according to their functional grouping. So this one corresponds to that one, obviously, and that with that, and that one with that one, and that with that. And this is what we get if we use k-means. So here are the 4 clusters of profiles using the full model, and this is what we get when we use k-means, without which we can do very quickly and very without any trouble. So. Now there's lots of these data sets, and these data sets by Spellman, they've been analysed to death in the literature, of course, but in this particular one we've got 612 genes, we've got 18 time points. I think they're measured at 7 minutes apart, and again we can just fit this model, and this time we come up with 12 clusters of the profile vectors for the genes. So just to summarise, the mixtures of linear mixed models are useful in trying to model biological processes, or any process where there's periodicity, or even if there's not, as in cross-sectional data, where you might take multiple samples at one time point. So they provide a flexible tool to cluster data. Well, not necessarily. Well, it can handle high-dimensional data. The data we've been looking at, because we only have had 17 or 18 time points, I suppose it's a bit hard to call that high-dimensional, although I think in the old days if we had a data set with about 20 variables we'd be tempted to call that high-dimensional. But it can handle structured data for a wide range of experimental designs, and longitudinal data where you've got replication or you don't have it, and cross-sectional data where you have, say, multiple samples in a class, you only have the one time point. And the other thing is, of course, you can incorporate any experimental design information that you have, and also the fact that you have covariates. They may be biological or clinical ones. So I think I'll stop there. Thank you.
What are the 4 functions? The reason for that question is, are you sure that none of those genes can have multiple functions amongst those 4?
Well, the actual categorization into those 4 things was done by Cho, and so one is I did remember what the 4 are, but I mean I haven't got a biological background, but they actually did the grouping into these 4 things, what they related to. But if I look through the paper, I could pull them out. But so I don't think they would have in this particular case the way they pulled out a subset with these, I think, 4 distinct functions. Yeah.
Was there a phase in your periodic function?
No, the phase was taken to be 0, because they use a temperature to stop the process. They can stop the cycle at any point in time and then start it up whenever they want to. And so all these things were started up they were started up to be in synchronous, to be synchronized. So
I think there are different checkpoint genes along the cell cycle, so some of them might be shifted along.
I think some of the genes, their product is needed for this, the life to progress, the cycle, and so if they, and they're temperature sensitive, so they can stop the cycle wherever they want to, and then restart it whenever they.
So you described a static approach for modeling this expression profile, and also you mentioned in the literature other work with static approaches, but there are other people that started to use temporal modeling indeed for clustering. Do you think that this is also a valid way to go, or do you have any feeling for that? Or?
What, modeling the effect of time?
With temporal model instead of static models.
I think people are moving towards the fact that taking into account the effect of time and the order in which the observations are being made.
Jeff, I've got one technical question. You said that the likelihood is not computable?
Well, I can't write it down in closed forms. Okay.
Isn't everything linear Gaussian? So?
No, I mean, if you've got correlated observations on the same entity, that's okay to handle, because that's a multivariate normal still. But when you've got correlated observations, it's just not a product of their density. So you can form one great big
isn't it just one
vector,
huge
but to actually work with, to put it in a form where you can manipulate it, it's...
Are the U and V's separable, or are they identifiable?
Sorry, what?
I mean, in the forms you have the terms U and V. Are they separable?
Are they separable? They're design matrices, but they can be specified independent of each other. In the design I had, used just a column vector of ones, and V is the identity matrix.
But from the data, you only have the sample covariance matrix, right? So you can have different U and V's that gives you the same sample covariance matrix.
Oh, I seem to. Okay. The model's identifiable. Is that what you're? No, it's identifiable with those. You have to be careful, of course, when you fit a constant at each time point. Then you can't include. If you didn't have the periodic function, the linear combination of cosine and sine curves, and you just fitted a separate constant at each time point, then you can't have the cluster random effects in there, because they wouldn't be identifiable. But you specify these design matrices so that the thing will be identifiable.
No more questions? And let's thank our speaker again.